So the next speaker we have is also going to refer to some interactions with between heterotrophs and cyanobacteria. This is Dr. Stephen Biller, and he is an assistant professor at Wellesley College, working on Prochlorococcus and looking at interactions with heterotrophs during extended periods of darkness. So take it away, Steve.
Thanks so much, Lisa. Thanks to all of you for being here, for your attention. It's nice to see a lot of new and familiar names on the list. So at the outset, I just want to take a second to acknowledge Allison Coe, who works in Penny Chisholm's lab at MIT, that was sort of an integral part of all the work I'm going to be talking about today. So just to remind everybody about my favorite cyanobacteria, Prochlorococcus. It's a small, free-living picocyanobacterium that's incredibly abundant in the global oceans. Anywhere in kind of mid-latitude waters, we can find different types of Prochlorococcus, global census populations on the order of 10 to the 27th cells. So they're an incredibly diverse group of organisms, a lot of fascinating stories coming out about them. And thanks to efforts of Lisa and many other people over the years, we also have beautiful culture collections of some of these different strains that we can play with in the lab to try and understand what makes them tick. So like all good cyanobacteria, Prochlorococcus's life is really driven by the day-night cycle. So we know that in the oceans, the cell division cycle is very closely tied to light input. Work from Eric Zinzer and others has also shown that the transcriptome of Prochlorococcus is very beautifully attuned to day-night cycles as well, with really significant fraction of the transcriptome showing these beautiful diel oscillation patterns shown here on this figure on the right. So our question was, well, what happens to photosynthetic cells when there is no light? And so I asked this not just because I like to torment these cells, but because it is ecologically important, because even though cyanobacteria like Prochlorococcus are really abundant in the surface euphotic zone of the oceans, there's been a number of types of evidence over the years that show that Prochlorococcus, it can be found up to thousands of meters deep in the ocean, well below where there's any, appreciable photosynthetic, photosynthetically available radiation. This can happen through a lot of different processes, different types of vertical waves and things. But cells can be brought down into the dark for potentially extended periods of time. So we were kind of curious, well, can they survive for that long? And so when they come back up, that they could kind of keep on their merry way, or are they pretty much just toast when they're down there? So Allison has been working on this for a number of years with experiments in lab cultures that go something like this. So here we're just looking at growth as measured just by fluorescence of these cultures in the lab over time. Notice that this is a log scale x-axis. We spent months on this. So if you just grow Prochlorococcus in a normal day-night cycle, they grow with sort of normal exponential growth characteristics. We now sort of put them in the dark for an extra day. What we see is that in cultures of pure Prochlorococcus, they really don't like that. The cultures, crash soon after, the fluorescence drops, and even when you look months later, these cultures just don't grow. The interesting thing is that if we now have another cell in these cultures, so if we add a heterotroph to these cultures, so they're not growing completely in pure culture, put them through that same process, now we see that the Prochlorococcus are able to regrow after a little bit of time. Still, the bulk of the population doesn't like it, but there is some subfraction of those cells that is able to survive and regrow once they come back into light. So this raises a whole lot of questions about how heterotrophs are helping Prochlorococcus in this kind of a situation. We know sort of this canonical autotroph, heterotroph kind of paradigm tells us that Prochlorococcus should be over here on the left doing photosynthesis, sending all of its fixed carbon, or at least some amount of it to the surrounding community to feed the heterotrophs. But it turns out the heterotrophs actually do a lot for Prochlorococcus as well. So one example of this is from work from Jeff Morris and Eric Zinzer have shown that the heterotrophs can actually play a really important role in mitigating reactive oxygen stresses to Prochlorococcus. But it seems like there was kind of more going on here. So we were really interested in kind of what else might be going on in these types of Prochlorococcus heterotroph interactions. So specifically, what I wanted to do was kind of look here at kind of this early time period when Prochlorococcus enters this sort of period of extended darkness beyond that normal day-night cycle, and try and figure out, well, what's happening to these cells at this point? How are they reacting? What can that tell us about what might be going on in these conditions? So we set up an experiment like this where we had cells of Prochlorococcus growing either in pure culture or in co-culture with one specific heterotroph, an Alteromonas strain. We put these cultures through either a normal light-dark cycle or kind of this extended dark period where they were growing in light-dark, and then the sun just kind of didn't come up at this point. So we took a transcriptomic approach to try and look at kind of what was going on in these cells. We extracted RNA, sequenced them. We have all the reference genomes. So we're able to align the reads and start trying to figure out what was going on. So one of the first things that I noticed, just kind of doing some basic QC on these transcriptomes, is that the heterotrophs actually played a really interesting role in stabilizing that transcriptome during extended darkness. So what I'm showing you here are these plots of individual gene expression just in kind of two of our replicates, where each dot is an individual gene. So we can see that under normal light-dark conditions, Prochlorococcus alone, all the replicates looked pretty much transcription like each other. There was pretty good solid replication of the expression patterns. Similar things when we had the Alteromonas in there on a normal light-dark cycle. So all the cells were kind of doing pretty much the same thing. But then now we sort of fast forward to after these cells have been in the dark for an extra 13 hours or so, and a lot of the Prochlorococcus gene expression patterns, in these individual replicates started looking quite different for one another. So again, these are bulk averages across lots of cells in the populations, but these cells growing alone really seem to be freaking out. And there's a lot of changes in the transcriptome dynamics. Whereas, there's the replication among the different bottles that we had in the co-cultured context, even in this extra dark context, they still were all kind of doing the same thing. So that was an interesting kind of first note. We sort of then look at, okay, what's actually changing? And so we just kind of look overall at, just how many transcripts in the Prochlorococcus genome are changing in our normal light-dark cells compared to these that are in extended darkness. When they're sort of in the normal night phase, the expected period of night, there was essentially no differences seen. Whereas even, a few hours after the sun didn't come out for these cells, we started seeing large changes in the transcriptomes of these cells. Both some genes that are increasing in relative abundance under extended darkness compared to light dark, as well as those that decreased a fair amount. There were some of these genes here sort of below these white dots that were pretty consistent across all the replicates, and then and also between these sort of co-culture versus azenic conditions. But we also had some gene expression patterns that were different changes when we had these pure cultures versus the mixed cultures. So my kind of involvement in utilization of some of the MetaCyc tools comes from trying to really figure out how to make sense of what these metabolic changes were. So we had some very complicated RNA-seq data sets to try and make sense of. So I turned to pathway tools to try and help me organize and interpret these data. So this is not the figure on left is not supposed to be anything you're interpreting directly. I'm sure it's pretty small and fuzzy on your screens. But in red are some examples of genes across many different pathways that were changing under different conditions in extended darkness. And so I used some of the pathway enrichment analysis tool capabilities in pathway tools just to kind of help make sense and organize and categorize some of these differentially abundant transcripts. So just for the sake of brevity, to kind of just broadly kind of categorize, what was happening in the dark, When we have cells growing in pure culture, we saw a lot of induction of various stress response pathways. Some of the light, some aspects of the light reactions of photosynthesis were increasing. They seemed to be increasing photosystem expression, trying their best to get any photon that might still be out there. They also seem to be switching to more of a fermentative type of metabolism. We also saw a lot of decrease in other pathways related to just general growth, including biosynthesis pathways. By contrast, when we look at the cells that were in co-culture, when they had this other heterotroph around, we saw, that they were still able to do a lot of biosynthesis. We saw expression of a number of pathways suggesting that they were trying to degrade different types of organic compounds. We saw increased utilization of heterotroph related transcripts, and also a number of other transporters that Prochlorococcus seemed to be turning on in co-culture that weren't on in the azenic cultures. So this leads us to think that maybe Prochlorococcus are really acting more mixotrophically in dark and sort of actually taking some organic carbon back from these heterotrophs and using those to try and survive in the dark. One last very brief story I'll tell here is about sort of those transcriptional rhythms and kind of the oscillations. So, I showed you earlier, we know that Prochlorococcus gene expression is really, tied to these beautiful day-night rhythms, and work from Liz Otteson and others is showing that sort of these transcriptions in the autotrophs in the ocean can similarly drive transcriptional oscillations in other heterotroph communities based on kind of the availability of this photosynthate that can allow them to go ahead and start growing and dividing. And so when we looked at this transcriptomic data, and I'm just here categorizing using statistical tools, sort of the percent of all transcripts that seem to show statistically significant periodicity. Prochlorococcus alone, showed pretty hefty proportion of the transcripts were periodic. Interestingly, that amount of, or that the number of periodic transcripts increased in co-culture, even in normal day-night contact light cycles. And we also saw a few transcripts that were seeming to be kind of periodic in our Alteromonas cultures, a portion of the culture as well. Now, this decreased quite a bit under extended darkness, where very few transcripts showed periodicity anymore in the azenic cultures. But in the co-cultures, there was still almost 25% of the transcripts that managed to kind of maintain those transcriptional rhythms, due to some types of aspects of interactions with the heterotrophs. So, we think that, there's lots of models of how these light inputs are, fed into Prochlorococcus to drive these transcriptional rhythms. Interestingly, Prochlorococcus doesn't have a complete chi-ABC oscillator. It's missing the chi-A component. So it seems, we're acting more of an hourglass model where these transcripts sort of have to be reset kind of every day due to light input, potentially acting through, either redox potential in the cell or ATP-ADP ratios or other signals. Seems like there is also a role for heterotroph metabolism and interactions in helping to drive some of these internal dynamics in Prochlorococcus as well, whether through organic carbon inputs, changes in that reactive oxygen environment, or some other components of all of that we still have yet to kind of figure out. So I'll stop there by just kind of summarizing that heterotroph interactions really impact Prochlorococcus stress responses and transcriptome dynamics. It looks like some of the compounds provided by these Alteromonas heterotrophs are able to sustain Prochlorococcus under extended darkness conditions, and that these types of biotic interactions can really influence these transcriptional rhythms. So I'm really excited about this CyanoCyc initiative and all the effort that's being put into this, because I think that there's a lot of role for improving kind of these metabolic databases to help us understand, make sense of these metabolic changes We have a lot of other kind of data, kind of, building off of this work. Turns out you can get mutants of Prochlorococcus that seem better able to handle being in the dark. And we're really interested in trying to interrogate some of that metabolism as well as these co-culture interactions in more detail. So I'm also very excited for the next talk as well. All right, so I'll stop there. And all this work was begun in Penny Chisholm's lab at MIT. Again, huge shout out to Allison Coe, who has been doing heroic, 6-month-long experiments on this for many years. Also thank my lab at Wellesley and some folks who are kind of picking up on various aspects of this work and funding sources as well. And thank you all.